Today, we're going to expand our horizon beyond division research. We have a fantastic scientist coming to give us her take on research related to, I would say, in general, to genetics. So that's wonderful that Joanna agreed to come and join us today for the seminars. As you perhaps also noticed, our building is coming up, and yesterday was official announcement of us moving into a new location once the building will be finished. So that's wonderful news. All right, with that, Yasmin, maybe you will introduce our speaker today.
Hello, everyone joining on Zoom. My name is Yasmin Solano. I'm a fourth-year PhD candidate in Dr. Philip Kaiser's laboratory. I have the pleasure of introducing to you all today Dr. Joanna Wysocka. Following her completion of graduate studies in Cold Spring Harbor Laboratory, Dr. Wysocka carried out her postdoctoral studies at the C. Davis Alice Laboratory at the Rockefeller University. Following this, Dr. Wysocka started her career at Stanford University School of Medicine, where she went from assistant professor to associate professor to professor within the Department of Chemical and Systems Biology. In this process, Dr. Wysocka became an HHMI investigator and the Lori Loki endowed professorship. Dr. Wysocka's success has earned her numerous awards since her early career, such as the Surley Scholar Award, Baxter Foundation Award, W.M. Keck Distinguished Young Scholar in Biomedical Research Award. Further into her career, she was awarded the AAA Harlan Winfield Mossman Award in Developmental Biology, the Walkhoff Chair Award, Momentum Award from the International Society for Stem Cell Research, and the Normus Foundation Research Award. She is an active member at multiple Stanford centers, such as the Comprehensive Cancer Center, BioX, to name a few, an editorial board member within Cell, Molecular Cell, Developmental Cell, and Trends in Genetics, and she is on scientific advisory boards where she currently is participating in the Kinship Foundation, Whitehead Institute of MIT, Surley Scholar. Her research laboratory is focused on understanding how regulatory information encoded by the genome is integrated within the transcriptional machinery and chromatin context to allow for emergence to form and function during embryogenesis and evolution, and how perturbations in this process lead to diseases. In the recent years, Dr. Wysocka's group has published 31 peer-reviewed papers, many in high-impact journals such as Nature, Nature Genetics, Science, and others. She has been cited 24,890 times as of April 2023. And she takes pride in providing a culturally diverse scientific environment with brilliant next generation scientists, some who have gone to be extraordinary faculty at premier universities within the United States, Europe, and China. So today, please join me in welcoming Dr. Joanna Wysocka.
Thank you very much for this very kind invitation, and I apologize I cannot be here in person today. Let's see. Do you see my slides?
Yes.
Great. So I was also really happy that somebody can pronounce my name correctly. That's rare. Usually I'm known as Joanna Wysocka, but I see you have a huge Polish contingent in the department, and that's probably why, including an old friend of mine, Dorota Skowrońska, who is, I believe, now a faculty member in your department. And we knew each other when we were both young and beautiful, and you can ask her about that. All right, so a pleasure to be here. And I want to start by saying that, perhaps a central, and maybe some could argue the central question in modern biology. I'm sorry, it's just, sorry. Okay. Perhaps a central, if not the central question in modern biology is to understand how information encoded in our genome reveals itself through the developmental process and through interactions with the environment to produce diverse human phenotypes. And in my lab, we're in particular interested in understanding how regulatory variation encoded by the genome gives rise to the phenotypic variation between species, so what makes us human, within the human species, what makes us individual, and how these regulatory changes can give rise to disease. So we use multiple model systems to understand this genotype to phenotype connection, but we think that one particular interesting model to study genotype to phenotype connection is the human face. So human faces are generally interesting, and they're interesting biologically from many different standpoints. So evolutionary recent changes in craniofacial complex allowed for emergence of uniquely human facial features and a number of biological adaptations. Within humans, face plays key roles in individual identity and in social interactions, and of course, is also a strong subject of sexual selection. And finally, craniofacial anomalies account for a third of all congenital diseases. And because of such important role of the face in social interactions and individual identity, these diseases have not only huge medical implications, but also devastating social consequences for patients and their families. So if we want to understand how human face forms, we have to go back early in development, where a very unique cell type is induced and specified. And these cells are called cranial crest cells. They arise early in development at the border of the neural plate. And then they undergo epithelial to mesenchymal transition. They delaminate, migrate throughout the body, and at their destination, they differentiate to a variety of cell types, including bone and cartilage of the head and face. So, of course, craniofacial development involves interactions of many cell types, but cranial crest cells are the major cellular component of the developing craniofacial skeleton. So these cells form and these processes occur really early in development in the first trimester of human gestation. So this makes it a rather inaccessible system to study in an in vivo context. So many years ago, my lab developed in vitro protocols that allow us for derivation of cranial crest cells from human embryonic stem cells or iPS cells. And we also have done this for non-human primates and especially for the chimpanzee to allow evolutionary comparisons between cranial crest cells of different species. And this in vitro derived cells, we validated them with numerous assays. They recapitulate a lot of the bona fide features of the embryonic neural crest isolated from model organisms. And as I say, over the years, we use this model system to study variation, to study disease, and this really allowed us to probe molecular mechanisms at the stages of human development that would be otherwise inaccessible. And in particular, many years ago, my lab published this comparative epigenomic study of human and chimp cranial crest cells, where we analyzed chromatin patterns in multiple individuals of both species, or rather cranial crest cells derived from them. And this allowed us to systematically identify enhancers or candidate enhancers that change in regulatory activity between human and chimp based on this quantitative comparisons of the epigenomic marks and characteristic chromatin features. So this systematically identified enhancers that change in regulatory activity. We were able to validate a number of these enhancers in vivo in mouse transgenic enhancer reporter assays. And also we were associating these changes in enhancer activity with changes in expression of genes, including many genes that were previously associated with facial morphology in model organisms. So that gave us a list of candidate genes whose evolutionary modulation likely modulated change in morphological features that occurred during recent human evolution. But because human and chimp genomes are so similar, this allowed us also for doing quantitative analysis of epigenomic features in relation to the underlying sequence changes. And through this analysis, we're able to identify sequence motifs whose gains or losses are associated with gains or losses of enhancer activity. And our null expectation for this analysis was that we'll identify motifs in key neural crest transcription factors, and gains of this transcription factor motifs will be associated with gains of enhancer activity during evolution. But we certainly did not expect to identify a singular motif that would be much more predictive of enhancer activity change than any other motif. And yes, this is exactly what we found. We found this long motif previously not characterized or not seen, that was most predictive of activity changes and also most enriched at this species biased enhancers, or enhancers with species biased activity. Because the motif was novel, we got to name it, and we called it coordinator, because at the time we hypothesized that whoever binds the coordinator will play an outsized role in coordinating enhancer activity in the neural crest. And that's why changes in this motif sequence are so tightly linked to changes in enhancer activity. So for a long time, I thought that this finding was one of the most interesting observations from our EVO-DEVO paper. And I tried to shop this project around the lab with a mixed success until Sang-Soo Kim, a really talented postdoc, entered the lab right before the pandemic. And he was really passionate about cracking the coordinator mystery, and he really did. And I'm really delighted to be able to tell you about it today. So first, it did not escape our notice that coordinator contains a homeobox, a motif bound by homeodomain transcription factors, and an e-box, a motif bound by BHLH transcription factor, separated by a fixed A-rich spacer. So keeping this in mind, Sang-Soo asked three simple questions. First, in which cell types is coordinator motif active? Then, who binds coordinator? And also, why is it so predictive of changes in enhancer activity? So first, in which cell types is coordinator motif active? To address this question, Sang-Soo analyzed over 2,300 DNA-seq and ATAC-seq datasets from various adult and embryonic cell types, both in human and in mouse tissues, and asked in which cell types is coordinator enriched in open chromatin regions. And we also required that coordinator enrichment was higher than enrichment of either of the constituent homeobox or e-box motifs. And with this analysis, coordinator turned out to be enriched in open chromatin regions of a surprisingly few cell types. So as expected, we saw enrichment in human cranial crest cells, and also in mouse facial tissues, which at the stages of mid-gestations are mostly composed of cranial crest cells. Sang-Soo also saw enrichment in limb bats and smaller enrichment in fibroblasts, which are mesenchymal cells of either mesodermal or neural crest origins, and neuroblastomas, which are cancers of neural crest origin. But all in all, out of these hundreds of cell types analyzed, coordinators were remarkably specific to the embryonic mesenchyme of the face and limb. So then, who binds coordinator? So BHLH and homeobox transcription factor families are among the largest transcription factor families encoded by the human genome, with literally hundreds of members. So when Sang-Soo asked which transcription factors can bind to the constituent e-box or homeobox of the coordinator, he saw that many transcription factors matched this have motifs. So this didn't really narrow our search too much because, of course, again, a lot of BHLH factors bind some version of the EBOX motif, and homeobox factors bind homeobox motif. But then Sang-Soo looked at the correlation of candidate transcription factor expression with the coordinator enrichment in open chromatin regions in all cell types in which we detected at least some coordinator enrichment. And now he saw that one transcription factor in particular, called twist1, was really nearly perfectly correlated with coordinator enrichment in different cell types. And we were excited about this because twist1 made a lot of biological sense to us. It's a key driver of mesenchymal identity in both mesoderm and neural crest. It's known to be required for both face and limb development, both in humans and in mice. And it's also been studied quite a lot in the context of cancer, as it's involved in epithelial to mesenchymal transition and cancer metastasis. So to confirm that twist indeed can bind at coordinator, a sequence Sang-Soo analyzed twist binding in mouse tissues dissected from E10.5 mouse embryos, from all the facial tissues and limb bud tissues in which we previously detected coordinator enrichment. He also looked at human cranial crest cells and at a number of cancer cell lines that expressed twist, including neuroblastomas, which express both twist and homeodomain TFs. And he also found one cancer cell line that expressed high levels of twist, but it nearly lacks expression of homeodomains. So this was to ask whether twist binding to a coordinator is dependent on the presence of homeodomain factors. So when he analyzed the data, he saw that the top 20,000 ChIP-seq peaks for twist were highly enriched in coordinator in cranial neural crest cells in limb buds, as expected, if twist is indeed this coordinator binding factor. We saw lower enrichment in neuroblastoma, but interestingly, in cells that expressed high level of twist, but no homeodomain factors, there was no twist binding at coordinator site. So this really was consistent with the idea that homeodomain factors may be required for twist to bind at coordinator. So which homeodomain factors this may be? So unlike for twist, where we saw perfect correlation of twist with coordinator enrichment across cell types, when Sang-Soo analyzed expression of homeodomain factors in coordinator-enriched cell types, he saw there wasn't one factor that was perfectly correlated. Rather, there were groups of homeodomain TFs that were highly expressed in subset of coordinator-enriched tissues in a manner that was dependent on the positional identity. So, for example, ALX factors are highly expressed in the upper face, and they're also known to be involved in upper face positional identity. DLX factors expressed in the lower face, and canonical Hox genes are expressed in limb buds. And more broadly, throughout the embryonic mesenchyme, we also saw expression of PRX factors and MSX1. So then to study mechanistically the cooperation between twist and homeodomain TFs at the coordinator, Sang-Soo went back to our in vitro model, and he generated a series of edited human embryonic stem cell line in which he endogenously tagged these transcription factors of interest with the FKBP dagron and V5 tag. And this allows us to differentiate these cells to human embryonic stem cells and to cranial neural crest cells, and then by adding dagron-inducing molecule, rapidly induce degradation of each transcription factor of interest, allowing for this acute perturbation. And I should say in our cranial neural crest cell model, twist is expressed at high levels, and also there are 4 homeodomain transcription factors expressed at high levels: ALX1, LX4, PRXX1, and MSX1. And this is consistent with the anterior facial identity, which we have characterized before. So first, Sang-Soo asked, where are these homeodomain transcription factor bindings? Are they binding together with twist? So to do so, he's done ChIP-seq or cut and run experiments with twist1, another key neural crest transcription factor AP2 alpha, and characterized sites that are bound either by twist only, AP2 alpha only, or by both, and then looked how association of homeodomain TFs looks at these sites. And he saw that homeodomain TFs are enriched at sites of twist occupancy, but not at the sites that were bound only by AP2 alpha. And moreover, coordinator motif was the topmost enriched motifs in this ChIP or cut and run ChIP or cut and run peaks for homeodomain TFs, indicating that in the context of cranial crest cells, these homeodomain TFs mostly bind in the context of the coordinator motif rather than in the context of the canonical homeobox motif. So then what happens when we degrade twist? What happens to homeodomain binding? So Sang-Soo has done this experiment, and now I'm showing you an example with ALX4. When we degrade twist, within hours, ALX4 disappears from chromatin, which allowed us to conclude that the twist is required for association of this the homeodomain factors with chromatin. Moreover, we also saw that when we degrade twist, there's a massive effect on both H3K27 acetylation and on chromatin accessibility as measured by ATAC-seq. So this indicates that twist really plays a key role in chromatin opening at neural crest cell enhancers, and perhaps can explain why this factor or why coordinator itself was so predictive for enhancer activity changes when this motif was changed during evolution. So now what about the reverse relationships? What happens to twist when homeodomain factors are degraded? So in this experiment, Sang-Soo degraded ALX and asked what happens to twist binding using ChIP assay. And he saw that binding of twist at the coordinator sites was diminished, but instead it was increased at the canonical targets of twist, which are EBOX and double EBOX sites. This effect is qualitative rather than absolute, but you should remember that there are other homeodomain transcription factors that also bind at coordinator that are still left in these cells in this experiment. So then Sang-Soo asked whether ALX4 would be sufficient to redirect twist to the coordinator sites in a naive cellular setting. And this naive cellular setting for us were simply HEC293 cells, which don't express twist and express low levels of homeodomain TFs. So Sang-Soo overexpressed twist in the presence or absence of ALX4 and analyzed binding of twist by ChIP-seq. And he saw that in the presence of ALX4, twist prefers coordinator, but in the absence, it prefers its canonical EBOX and double EBOX sites, suggesting that homeodomain TF, like ALX4, is able to switch twist specificity from its canonical recognition motif towards the coordinator. ALX is not the only factor that does it. Sang-Soo tested a number of other homeodomain factors expressed in the neural crest, and they're all to some extent able to redirect twist to coordinator sites, but they do so with differential efficiency, with ALX4 and generally ALX factors being the most robust in being able to direct twist towards the coordinator. So this explains why coordinator is active in the developing face and limb mesenchyme, because these are the cell types where both twist and homeodomain transcription factors are co-expressed at high levels. However, here's a conundrum. There are many other cell types in which other BHLH factors and homeodomain factors are co-expressed. And yet in these cell types, and this is particularly prevalent in the brain, we don't see coordinator enrichment in open chromatin regions. So this suggested to us that there is really something unique about twist that is able to promote its cooperativity with homeodomain transcription factors. So for a while, we considered that perhaps twist uniquely is able to directly physically interact with homeodomain TFs in solution. And Sang-Soo has done a number of assays, but was never able to confirm strong interaction between these factors in solution. And instead, we saw very strong interaction with TCF factors that are BHLH factors that are known, that are broadly expressed, are also known to heterodimerize with twist to bind an E-box. So no direct interactions in solution. What else can it be? There's another mechanism by which certain transcription factors can cooperatively bind juxtaposed DNA sites that are arranged in specific orientation and distance, but without forming stable direct protein-protein interactions. And perhaps the most well-known example of this is OCT4 and SOX2 binding at the composite motif in pluripotent cells. And this mechanism is referred to as the DNA-guided or DNA-mediated cooperativity, because this contact between transcription factors can occur upon binding or engaging the DNA. Therefore, DNA sequence guides the cooperative interaction. So we were wondering if this type of mechanism may be operating here, and we collaborated with UC Taipale, and Katia Morgunova, a senior scientist in UC's lab at the Karolinska Institute, crystallized TWIST1 and TCF4, its heterodimerization partner, and the LX4 DNA binding domains that are co-bound to the consensus coordinator motif. And as expected in this structure, we saw that the TWIST TCF4 heterodimer recognizes the E-box portion of the coordinator, LX4 recognizes the homeobox portion of the coordinator, but importantly, ALX and twist loops, this sort of flexible loops that make weak contact when the factors are co-bound at the DNA. So it's not an extensive protein-protein interactions. You can think of it as these proteins just kissing each other when they're co-bound on the coordinator motif. So the prediction from the structure was that perhaps it's this loop contact, this weak loop contact, that provides this enormous specificity or selectivity for twist to cooperate with homeodomain TFs. And to test this, Sang-Soo went back to the overexpression assay. And now, in addition to testing wild-type twist, he also tested several versions of twist. First, he tested a version of twist in which this loop that makes contacts with ALX in the structure was replaced with a loop from a number of homeodomain factors. I'm just gonna, for a number of other BHLH related factors binding to the same E-box motif, I'm just gonna show you one example with NeuroD. And he also tested a number of single amino acid mutations within the loop. I'm also just gonna show you one example for simplicity. And as expected, wild-type twist bound at coordinator sites in the presence of ALX4, but neither the replacement of loops nor the single point mutations of loop were able to cooperate with ALX4. So this is consistent with the idea that the loop contacts is really, this wimpy loop contact is sufficient to provide enormous specificity for this cooperativity. And just as a control, I should add that all mutants transcription factors can robustly bind to E-box and double E-box. So we're not just generated a defective BHLH factor, but rather what was specifically lost was this cooperativity. So in other words, to summarize this part, coordinator guides cooperative binding of twist and homeodomain transcription factors in a manner that is dependent on the features of the twist loop. And through gel-shift assay that I won't have time to go over today, Sang-Soo also showed that in addition to the homeobox and E-box, the spacer both length and sequence matters for this cooperativity. But now, what might be the biological consequences of this cooperativity? I already told you that twist plays key roles in mesenchymal identity, and LX factors play key roles in positional identity in the phase. But when Saxu looked at the transcriptional effect upon loss of twist or ALX factors, he saw that many of the affected genes overlap and that expression of both cellular and positional identity downstream genes is affected in both. So this suggests, and without going into specific details, this suggests that at least one function of this cooperativity is to integrate cellular and positional information and gene expression programs for cellular and positional information. We also wanted to go back to this question of what may be the impact of coordinator binding TFs and their genomic targets on human phenotypic variation. This is, after all, how we first discovered coordinator, is through these evo-devo comparisons of enhancer activity. So unfortunately, we could not look at this question directly between human and chimp. I wish we could ever do chimp GWAS, but that's not realistic. But we could really tap into this enormous individual level variation that is observed within our own species. And over the years, we've been involved in a group of with a group of human geneticists in performing GWAS studies that overall uncovered over 200 loci that are associated with normal range variation in facial shape among human individuals, and different loci, these are mostly non-coding variants, I should add, are associated with variation of different aspects of the facial shape, or the jaw, the forehead, nose, and so on. And we previously also showed that facial shape heritability is enriched in enhancers that are active in cranial crest cells. But now we could ask a more specific question, which is facial shape heritability enriched selectively within this coordinator-containing and twist-dependent enhancers? So to ask this question, Zahin Naqvi in my lab looked at distal attack peaks that are changed upon acute degradation of twist. And he grouped them into the peaks that were upregulated or downregulated upon twist degradation. And I should say that it's these downregulated peaks that are selectively enriched in the coordinator. And he also compared them to a match set or ataxic accessibility match set of other open chromatin regions in the same cell type. And as you can appreciate from this plot, facial shape heritability was highly enriched in this coordinate-containing and twist-dependent distal ataxic sites, most of which were actually enhancers. And this enrichment was even higher than what we previously observed for the cranial crest cell enhancers overall. So this suggests that genetic variation in the coordinator-containing and twist-dependent enhancers modulates human facial shape. But what about the genetic variants at loci encoding coordinator-binding transcription factors themselves? We observed that each of the coordinator-binding TFs, so twist, ALXs, MSX1, and PRX, are associated with non-coding variants that were linked to the facial shape changes in human individuals. But the strongest effects and the most correlated effects were also associated with non-coding variants at transcription factor loci that were also the strongest coordinator binders, so twist, LX1, and LX4. So this suggests that quantitative changes in expression of this transcription factor themselves in trans can influence facial shape as well. And by the way, we're likely not the only species in which transcription factor coordinated binding transcription factors influence facial shape because allelic variants at the ALX1 locus emerged as those associated with beak shape variation in Darwin's finches. This is one of the leading locus associated with beak shape variation in this iconic species. So to summarize this part of the talk, I've shown you that coordinator and its transregulatory complex mediate phenotypic variation, and likely both through cis and trans mechanisms. So you may be wondering, well, how is this may be related to the cell types that I'm interested in? Do other cell types have their own coordinator-like motifs? So of course, coordinator itself is highly enriched or highly specific for embryonic mesenchyme of the face and limb. But our collaborator, UC Taipale, is done systematic analysis using in vitro Celix assay on the in vitro co-binding of different transcription factor pairs. And what he observed is, in fact, many different transcription factor pairs can show this cooperative binding, usually with a preferred single spacing. And yet only a handful of these interactions have been previously reported in the literature. So this suggests that this DNA-guided cooperativity may in fact be a very common mechanism of mammalian gene regulation, and that perhaps this composite motifs are very cell type specific and are hiding in some of our ataxic data and ChIP-seq data from specialized cell types that arise during development or in tissue homeostasis, and this is perhaps something that is worth considering. All right, so now I'm going to switch gears to the second part of my talk, which really arises from this question that we asked, whether there that we got interested in, whether there are shared molecular pathways that affect both disease-associated and normal range facial variation. And I already told you that the normal range facial variation is associated with non-coding variants. These variants are enriched with enhancers, including this coordinator enriched enhancers that we just discussed about. But what about the Mendelian diseases? In Mendelian diseases, often the observed mutations are coding instead of non-coding. But interestingly, in the face, these are quite commonly heterozygous loss of function mutations, suggesting that there is really a huge role for haploinsufficiency in craniofacial disease. And there's a huge number of these diseases. There are over 500 Mendelian craniofacial syndromes. These diseases are individually rare, but collectively very common. So we ask a very simple question. What happens if we intersect the predicted target genes of the GWAS non-coding variants with the genes that are mutated in Mendelian disease? Will we see any overlapping trends or pathways? And the only significant association that we got was with a single molecular function, and that is sequence-specific transcription factors. So what this suggests is that change in transcription factor dosage may be a very prevalent mechanism that is underlying facial variation in health and disease. And again, in the syndromic Mendelian diseases, it's often haploinsufficiency of the transcription factor, loss of function from one allele resulting or predicted to result at least in 50% change in dosage. And it's a dirty little secret in the GWAS field that this non-coding GWAS variants are in fact causing or associated with even more subtle changes in dosage. They usually effect size are on the level of 10, 15, 20% on gene expression from the non-coding variant. So even less than 50%. So this really got us thinking, how are such small changes in transcription factor dosage interpreted at the molecular and cellular levels, and how do they give rise to the phenotypic specificity? Because indeed, there are many transcriptions. Now we have nearly 30 transcription factors that are associated both with Mendelian disease and with normal range facial variation, but they all have distinct phenotypes. There's a huge phenotypic specificity in these dosage perturbations. And we decided to start tackling this question with our first example, or one example of this dosage-sensitive transcription factor interface, and that's SOX9. So for a long time, it was known that SOX9 is dosage sensitive for the facial development. Loss of function in mice is associated with broad craniofacial phenotypes and embryonic lethality. Then loss of haploinsufficiency, or we also showed a few years back that also loss can also be caused by loss of key craniofacial enhancers at SOX9. So 30 to 50% reduction at SOX9 level is associated with disease called Pierre-Robin sequence, which is primarily associated with lower jaw hypoplasia. So it's a disease, but with very specific manifestations in the face. And even smaller reduction SOX9 level is associated with normal range facial variation in the GWAS studies. And we formally shown, again, a few years back, that in mice, even 10 regulatory mutations that lead to only 10–15% reduction in SOX9 dosage, this small perturbation is sufficient to result in subtle but measurable changes in lower jaw shape. So overall, SOX9 is highly sensitive to dosage over a very broad range. So to start addressing the molecular mechanism of this, Sahin Naqvi in the lab developed a system for precise modulation of SOX9 dosage in our cranial crestal system. So again, we went back to this Degron, FKBP Degron system where SOX9 locus is endogenously tagged with FKBP Degron and amnion green. And now modulation of the DTAG concentration of this Degron-inducing molecule concentration allows us for very precise titration of SOX9 levels. And this is illustrated here by measurements of amnion green by FACS, or quantified as changes in SOX9 dosage. And this is shown as a percentage of the wild type dose. So then with this system for precise modulation, Sahin then could ask what are the dose to phenotype curves at the multiple levels of scale. So how does this titration affect, sorry, my computer is acting up today. How does it affect chromatin states, gene expression, cellular phenotypes, and ultimately morphology? So he first done this titration, SOX9 dosage titration in cranial crystals, and then measured chromatin accessibility by ataxic and gene expression by RNA-seq. And from these experiments, he identified distal ataxic sites that are dependent on SOX9, and those we defined as those that change significantly their accessibility upon complete degradation of SOX9, and there are about 35,000 of such sites. And I will refer them for simplicity as enhancers, but technically there are distal ataxic sites, most of which are enhancers. And similarly, he also systematically identified SOX9-dependent genes as those that change expression when we completely degrade SOX9. And for each of those dependent enhancers and genes, then he characterized dose response curve based on the titration experiment, and then fit it with two simple equations, either linear equation or the Hill equation. So asking whether this dose response is linear or nonlinear. And it turns out that most SOX9 regulated enhancers and genes respond nonlinearly to dose. So the fit to the Hill equation or nonlinear equation is better than fit with a linear equation. And then from this Hill equation, he would then extract two parameters, ED50 and Hill exponent. And for the purpose of this talk, I want you to focus on the ED50, which is defined as the SOX9 dosage at which either enhancer or gene reaches half of its maximal openness or expression. So in other words, you can think of it as this ED50 parameter as the quantitative measure of sensitivity, and the higher ED50 values, the higher sensitivity of the enhancer or gene to SOX9 dosage. So that's all you need to know. So just to give you an example how this data looks like on individual enhancer level, most SOX9 regulated enhancers and genes are in fact buffered against small to moderate changes in SOX9 dosage. So these are changes in ataxic signal upon titration of SOX9 dosage, and shown here as a wild type, as a percentage of the wild type dose. And as you can see, having the dose to 50% doesn't really have much effect on this enhancer's accessibility. And most enhancers and genes that are regulated both by SOX9 behaves this way. But there is a subset of enhancers and genes that are actually highly dose sensitive, and they start losing accessibility or expression immediately after you start diminishing SOX9 dose. And we also show that the sensitive enhancers are in fact enriched for specific DNA sequence features. We're still trying to fully understand the sequence features that dictate sensitivity of enhancer to dosage. And moreover, through modeling, computational modeling, Sahin also showed that this enhancer responses can partially predict gene responses. But then, what are these sensitive genes? What cellular processes are these dosage-sensitive genes are involved in, and are they in some ways associated with phenotypes that are relevant for the variation and disease? So early on in the project, Sahin noticed that multiple prochondrogenic genes are among the dose-sensitive genes. And this was interesting because some of you may know that SOX9 is required for entry of cranial crest cells into a chondrogenic program, and is also considered to be a master regulator of chondrogenesis. So, excuse me, to characterize this more systematically, Sahin looked at all SOX9 regulated genes and first grouped them into two groups based on whether they are or are not associated with chondrogenesis. And then within each group, he divided them further to those genes that are downregulated, upregulated, or not changed upon differentiation of cranial crest cells to chondrocytes. And what he observed is that the genes that were both involved in cartilage development and upregulated during differentiation of cranial crest cells to chondrocytes, those genes had significantly higher ED50 values, indicating sensitivity to SOX9 dosage. So then we ask whether chondrogenesis itself is sensitized to SOX9 dosages as well. And again, Sahin did the titration experiment, but this time during CNCC differentiation to chondrocytes, and at day 21 of this differentiation, he measured mature cartilage formation using a colorimetric assay. And he saw that even though median dosage curve of cartilage formation is nonlinear, it actually much more closely resembles the curve for prochondrogenic genes, those sensitized genes, as opposed to the curve that we've seen for all other genes or all other regulatory elements. So this is really consistent with the fact that chondrogenesis itself is sensitized to SOX9 dosage, and this is likely arising through the sensitivity of the prochondrogenic genes. So then, can we link these dosage responses and morphology, both in the disease context and normal range variation? So Sahin went back to the SOX9 regulated genes and divided them into different categories based on their association with craniofacial disease. In the first group, in the purple group, were genes that are regulated or dependent on SOX9 and that also show similar phenotypes as seen in Pierre-Robin sequence, namely the hypoplastic lower jaw. He also looked at genes that are associated with other unrelated or not phenotypically related craniofacial disorders that showed either recessive or dominant genetics. So with recessive, you need to have both copies of gene mutated to have disease. With dominant, a single copy can be mutated for disease, and many of the haploinsufficiencies are in fact in this dominant category. And the fourth group were the genes that were regulated by SOX9, but not associated with any craniofacial disease. And when he looked at the ED50 value, he saw that the genes, the purple genes, the ones that are associated with overlapping phenotypes at the SPR sequence, were in fact the most sensitive to SOX9 dosage. But interestingly, the green genes, so the ones that many of which are also dosage sensitive in the face, but associated with distinct phenotypes, were actually highly buffered. They had very low ED50 values. So this suggests that most of the key craniofacial genes are in fact buff, even though they may be regulated by SOX9, they are buffered to small changes of the SOX9 dosage. But only the genes that act on the same phenotypic axis are in fact highly sensitive. And then we were interested if we can extend this paradigm to the normal range facial variation. And we again collaborated with Peter Claes, our long-term collaborator for facial genetics. And Peter obtained 3D facial scans from PRS patients and healthy controls, and that allowed him to define face shape changes along the healthy to PRS axis, and we'll refer to this as endophenotype. And then he scored over 8,000 healthy individuals for face shape changes along this endophenotype, so from the PRS-like to the normal-like, and performed GWAS analysis on this endophenotype. So we were very pleased to see that SOX9 is one of our top hits in this GWAS, but there are also many other variants that were recovered, most of them non-coding. So we can then identify SOX9-dependent enhancers that are in linkage with this PRS endophenotype variants, and then compare them with SOX9-dependent enhancers that are in linkage with other facial shape GWAS SNP with unrelated phenotypes, which we had from the previous GWAS analysis that I already mentioned. And again, what we saw that the variants in LD with endophenotype SNPs had higher ED50 values, higher dosage sensitivity than those with unrelated phenotypes. So the paradigm extends to the normal range variation. So just to summarize, we've done quantitative analysis of effects of SOX9 dosage and saw that most enhancers and genes are buffered against small changes in transcription factor dosage. But a small subset of enhancers and genes is highly sensitive to dosage. And it's these, what I will refer to as effector genes, in our case, these were prochondrogenic genes, that form a regulatory bottleneck that then transmits the phenotypic effects. At small changes in transcription factor dosage, such as those associated with common non-coding genetic variants at the SOX9 locus, we only see small degree of sensitivity for this effector genes, resulting in normal range shape variation along the healthy PRS axis. When SOX9 dosage is perturbed further at 50 to 70% of normal dose, then the effects on the effector genes get exacerbated because these genes are progressively more affected by the SOX9 dosage. And this is enough to lead to disease, PRS sequence, and this disease has highly specific manifestations in the lower jaw. Importantly, other craniofacial genes, and there are many, that are regulated by SOX9 but have unrelated phenotypes, are buffered against this degree to SOX9 dosage perturbation. And only when SOX9 is completely lost, these craniofacial genes are broadly perturbed, leading to broad craniofacial phenotypes and embryonic lethality. So a couple of final thoughts to end this. So we began to think that understanding such dosage effects and associated regulatory bottlenecks will be absolutely essential for comprehending human traits and disease. You know, we're all mutants, but we're all non-embryonic lethal mutants, and really understanding these dosage effects will help address where the specificity is coming from in human disease and in human phenotypes. And another point is that this model actually reconciles what we know from developmental biology, which is that developmental processes are generally very robust to perturbation. This definitely extends to transcription. There is a lot of redundancy in transcriptional regulation. And yet we have obvious observation from human genetics that many genes are dosage sensitive. So in our model, of course, most of the system is highly robust to perturbation, but there are these regulatory bottlenecks then that result in the dosage sensitivity. And with this, I want to finish and thank people involved. And in particular, I've focused on the work on Sang-Soo Kim, who is a real hero of the coordinator project, and he's on the job market this year. And also on the work on Sahin Naqvi, who has done all the dosage sensitivity exploration, and he's soon moving on to his own independent faculty position. And our EVA-DIVA project started from Sarah Prescott's thesis work, and she's now assistant professor at MIT. And I want to also acknowledge our fantastic collaborators, UC Typaler, Peter Clays, and Jonathan Pritchard. Thank you very much for listening. I'll stop now.
Thank you very much. Let's start with Dr. Swarup.
Yeah, hi, Joanna. This is Anand Swarup. I'm at NIH and Eye Institute. I've been following your outstanding work for the last few years, particularly your recent papers. And actually, Sang-Soo Kim was one of the top candidates in the STAT research that I was co-chairing. So I think if he's interested in eye research, ask him to connect to us if you have another person like that. The question that I have, I mean, in following your papers and reviews, and particularly your focus on doses, transcription factor doses, which is so important, and many people had ignored that earlier, and they just sort of say, okay, expression and this or that. But your work, and also on the DNA-guided cooperativity, is just so beautiful. I have not seen something that I would like. I was wondering, I've been thinking about it, but I don't know how to incorporate that in some of these models. And that's the kinetics of binding. So what you are talking about is the strength of binding, but the kinetics of binding, not only how strongly it binds, but how it dissociates, and whether the cooperativity, how does cooperativity come into play? Because DNA, something has to bind to DNA first. And what is the role of maybe post-translational modifications through some signaling or genome topology in some of this binding that I think I haven't sort of seen in your reviews yet, but I'm sure you have been thinking about it, and I wanted to sort of hear your thoughts. You know, if you have some insights to share.
Yeah, you're absolutely right, and this is the question that we haven't addressed, and you know, a lot of the field hasn't addressed, because of course this requires yet another scale of observation. So even though, you know, our perturbations, for example, are on the scale of hours, which is, you know, already sort of a step up in terms of that time resolution and so on. Really to look at the kinetics of binding and so on, which, you know, some of this process of association and dissociation occur on the scale of seconds or even shorter for transcription factors. This really requires single molecule tracking experiments, and we're definitely, you know, thinking about those experiments and started some collaboration to go this way, because you're absolutely right that our model where the coordinator motif guides cooperativity essentially relies on, and we showed through gel shift assay that both of the transcription factors, need to be bound to DNA for the cooperativity to occur. So the model is that they're essentially searching their DNA space, and they will occupy for longer periods of time motifs that are actually correspond to their cognate DNA binding motifs. But once they encounter this cooperative motif, like the coordinator, then through this loop interaction, the binding will get stabilized, and their occupancy on the site will be much longer than compared to individual motifs. So that's our model, and we're trying to test it using some single particle tracking experiments. They're very challenging, and it will still take it really requires taking it to yet another time scale of observation, and we've encountered some challenges with that.
Yeah, so there have been some in vitro experiments done by Barry Knox in retina field, where he took two transcription factors, CRX and NRL. CRX is a homeodomain and NRL is a leucine zipper protein, and they bind to a promoter element, say in rhodopsin region, and CRX can bind very quickly. Again, by single molecule tracking, you can see the time codes, but it doesn't stay there.
Yeah.
And whereas if NRL, which cooperates with CRX, is there, that's a slow binding on its own. When both of them are together with the DNA, then one stabilizes the binding of others, and the cooperative is there. But some of these are very in vitro experiments. We don't know how much of what is available in vivo. So it's not only the concentration, most genes and most proteins are present in very high amounts when we do that in vitro, but in vivo, it's also availability of the protein at a given site.
You're absolutely right. So the idea would be to try to do some of these particle tracking experiments in cells, while you can also vary the protein concentration at physiological levels. Yes, that's sort of the dream experiment.
Yeah, beautiful work, and I've been following your nice reviews. Your papers are very insightful and
thank you. I really appreciate it.
Very nice.
All right, now I have to mute my brother Anand. Oxygen. Dr. Brown.
Oh, yeah, thank you so much. This is Andrew Brown from UC Irvine. I'm a retina specialist and engineer, so I apologize if this is a naive question, but as in the eye, the neural crest population kind of emerges at the front of the eye and forms the stroma of the cornea and the endothelium of the cornea, as well as the anterior component of the iris. And there are disease processes like angle closure glaucoma, where the morphology and the relationship between the iris and the cornea can predispose someone to angle closure attacks with elevated pressure inside the eye. So I guess my question is, do you know if the neural crest populations that form the anterior segment of the eye are similar or the same as the cranial neural crest population?
No, we actually don't. We haven't characterized this population in depth. There's, of course, periocular mesenchyme that is crest derived, and there, we do think that there's a lot of sharing of we actually think that coordinator, for example, plays an important role there, and in fact, LX factors, for example, have very clear phenotypes in humans associated with perturbations of periocular mesenchyme, but in some of those, we haven't had an opportunity to really look at these more finely defined neural crest derived populations that you're mentioning.
Thank you.
Sam?
Hi, I have a quick question. What is the sequence conservation between the chimp and human twist? Does the chimp twist also bind to this same coordinator motif? And if you change a chimp enhancer sequence to become the human sequence, what happens?
Yeah. So good question, and that sort of allows me also to clarify something that maybe some people got may be confused about, which is that the changes in coordinator between human and chimp are symmetric, meaning that we see as many gains of coordinator in human enhancers as gains in chimp, and we also see losses in human and losses in chimp. And always the gain of enhancer activity goes with a gain of coordinator motif, and loss goes with a loss. So it's not like this is a human-specific motif or chimp-specific motif. It's just that because it has such an outsized role on enhancer activity in the neural crest, this is why, you know, whenever evolution tweaks with this motif, you actually see changes in regulatory activity. And yes, we actually looked at, and when you put back, you sort of then let's say you have a loss of enhancer activity in chimp compared to human associated with loss of coordinator motif. If you then optimize coordinator motif again in the chimp enhancer, you see commensurate gain in activity. So it's not only predictive, but we actually showed that through testing some of these enhancer variants that change during evolution in massively parallel enhancer reporter assays, we actually saw that this sequence changes are directly causative.
Dr. Skowrońska-Krawczyk.
Hey, Joanna.
Hello.
As usual, and you know, I loved reading your papers and listening to your talks, and it's always lots of questions and answers. I have two questions. So one is related to a little bit what Dr. Swaruk said. Well, actually, I was thinking, do you have any idea if there is a pioneering factor or pioneering situation that actually opens coordinator sequence, or twist maybe is a pioneering factor? I'm not sure about that. And another thing comes from a really long time ago, my work on the BHLH factors, when I noticed that there's possibility of some loop in BHLH factors in the loop region. Some amino acids can interact most probably with DNA. Do you observe or do you see anything like this just by you know, positively charged amino acids interacting with negatively charged?
Oh, you mean in extended motif outside of the e-box?
Actually in the loop, exactly.
In the loop, okay, no, in the loop, no. So we don't, just to answer this question, we don't see loop itself contacting the DNA at all. But regarding the pioneering, yes, I mean, all our circumstantial evidence suggests that twist should, with, you know, probably in some form with ALX, but the twist should be a pioneer in the system. We trying to you know, look at that and whether twist can bind, you know, can bind nucleosomal DNA, how it can bind nucleosomal DNA. We're trying to look at it. We don't have any data yet, but yes, the circumstantial evidence we have suggests that the twist may be a pioneer in the system.
Thank you.
All right, so let's just finish with two more questions because we have to go. We have to let Dr. Wysocka go to other, I'm sure, important meetings. So Zach and Vladimir, Dr. Kefalo.
Thank you so much for that really interesting talk. So I had kind of a conceptual question. So is there a strict spatiotemporal overlap of ALX1 and TWIST1 transcription factors during development? So are they kind of simultaneously up and down regulated together? Or is there data to suggest that there's kind of this domain specificity switching that's a normal part of development for TWIST1?
Yeah. So they come up together during, you know, in once as neural crest acquires mesenchymal identity in the cranial region, both twist and ALXs, and you know, depending on also which positional identity in the face. So in the upper face will be ALXs, in the lower face will be DLXs, but these transcription factors sort of come on simultaneously alongside with mesenchymal identity. However, at later stages, and generally, you know, what we see that developmental overlap of expression domains of twist and ALX really is, this is where what defines coordinator activity. But there are other circumstances, in development and outside of development. For example, in breast cancer EMT, twist is expressed, but we don't think that homeodomain factors play a key role. Or, in some later stages of craniofacial skeletal development, twist goes down, and in some cases, some of the homeodomain expression is still maintained. So basically, the coordinator activity occurs only when those two temporary are co-expressed and coincide, and this is essentially in this undifferentiated mesenchymal tissue of the face and limb, and that's why we think we see the coordinator activity and this cooperativity there. This is not to say that, of course, we tested already 2,300 different data sets, but of course, it could be that we missed some really important developmental populations that co-express twist and homeodomain factors, and those two could have coordinator activity. But as far as we can tell, the major areas of temporal co-expression for these factors is face and limb development.
Thank you.
Vice Chairman.
Hi, Joanna. This is Vladimir Keplov from UC Irvine. Beautiful work. Couple of questions. Can you explore the differences between males and females in the context of your transcription regulation network? And do you expect discrete differences, or do you expect more of a continuous spectrum of more subtle differences between males and females? And
between males and females.
Yes.
Yeah.
And my second question is, I think that the face symmetry is considered as one of the things we perceive as beautiful. So have you explored whether there are subtle differences in those transcription regulation networks between the left and the right side of the face, and whether they contribute to the level of symmetry of the face?
Yeah. So regarding the sex differences, yes, so this is really interesting. So first of all, a lot of sex differences in the face arise at puberty in response to androgens and so on. And this, our model, we don't have yet a good cellular model to really model this. That said, there's actually already sexual dimorphism in the face at birth that is detectable, and it's probably associated with prenatal exposure to testosterone. So we have done at GWAS on facial perceived masculinity or femininity. So you can, define again endophenotype for the feminine for the feminine versus masculine face, and then ask what variation in the population affects that aspect. And what we saw was that mostly the same loci that affect individual level variation can also contribute to this masculinity or femininity of the face. But we also saw a couple of peaks that are unique, which are probably interesting candidates for exploring in terms of this response to androgens and so on. So that's regarding that question. And regarding facial symmetry, we haven't looked at it, but I think it's a very interesting question, and one would expect. So facial symmetry is sort of thought to be a measure of developmental robustness, and this is perhaps also could provide sort of evolutionary rationale of why we perceive symmetry as beauty and why it could be selected for. Because in fact, perhaps it selects for the developmental robust, you want to mate with a partner that is developmentally robust and that has developmentally robust genetic programs. So I suspect that what is associated with facial symmetry is actually going to be different than genes associated with individual level variation. And that these are going to be there's clearly going to be environmental component to robustness, and that, you know, essentially in certain environmental conditions, certain, you know, stochastic mistakes could be more or less frequent, but that there is also probably a genetic component to this buffering or robustness. And there, I would expect actually to perhaps you know, different loci or different genes and different mechanism to contribute to that, but we have not yet explored this. But it's a very interesting question.
Thank you very much. And Joanna, do you see all of the panels of pictures? Do you see a Millen's dog?
Sorry,
There is a dog picture of Millen's, Dr. Millen's dog on the panel.
Dog?
Yes.
Or do I know what's
No, but do you have the panels of those pictures of us and the dog and other people?
Participants.
I don't know what you see on your
Zoom
screen.
participant panel.
Oh yeah,
Chris is teasing
yes,
Rob Mullins.
I see Robert Muller. Okay, yes,
Yes,
I see.
so
A lot
really
of
fundamental question is why his dog is so beautiful.
yeah, I mean he's cute, I have to say, right? And but also like this is dog has actually craniofacial mutations, which cause the I mean that's right. I mean because it caused the flattening of the face. This as humans, I mean I myself have cats with craniofacial malformations that flatten their face, and we as humans perceive that as beautiful because it's or like very appealing and very childlike, because these are, you know, big eyes and flat face. We really perceive this as a baby, and that's why the, you know, all the flat-faced dogs and cats are